Hello friends, I am Dr. Ujjwala Desai, working as an assistant professor at PS Modern College of Pharmacy, Nigri, Pune. So today we will study the acid-fast staining technique. So following are the requirements for acid-fast staining technique. First is your clean glass slides, then inoculating loop, which is also called as microwire loop, Bunsen burner in order to sterilize the inoculating loop, bibulous paper, that is same as that of tissue paper or filter paper, microscope, lens paper and lens cleaner in order to clean your objective and eyepiece, immersion oil, which includes either white liquid paraffin or cedar wood oil, distilled water, and 18 to 24-hour cultures of organisms. ZNN reagents. So in this Zeal-Nielsen method, the acid-fast staining method is also called as Zeal-Nielsen method. So following are the reagents which we'll be using for this Zeal-Nielsen or ZNN reagents. First is your primary stain, which usually includes carbon fission. So carbon fission is basically a basic fission stain, but in combination with phenol. So when the basic fission stain is used in combination with phenol, it is called as carbon fission stain. Decolorizer, it can be either only acid or only alcohol or an acid alcohol solution. So sulfuric acid can be used as only acid, alcohol, ethanol can be used, and acid alcohol solution in combination can also be used as a decolorizer. In secondary stain, we usually go for methylene blue or malachite green. So the basic principle involved in acid-fast staining technique is the acid-fast bacteria, for example mycobacteria or Neisseria, they contain mycolic acid as special component which is present in their outer membrane or cell wall. It is waxy in nature and due to which the normal stains cannot penetrate through the cell wall and hence they cannot be stained by normal staining techniques or gram staining techniques. They are resistant to staining with aqueous based stains like your gram stain technique. So, the primary stain, that is carbol fission, is lipid soluble, and when applied to the cells under heat and phenol, it helps the stain to penetrate deep into the waxy surface of the acid fast microorganisms. The excess stain is then removed with the treatment by acid ethanol reaction, and then a secondary stain is applied, for example methylene blue, in order to stain the non-acid fast bacteria. Now here you can see the procedure involved in acid fast staining technique. So first of all, you will sterilize the microwire loop by holding it in red hot flame for 2 to 3 minutes until it turns red hot. Then you will insert that microwire loop in the test tube containing inoculum. Then the loophole of that inoculum will be transferred to the glass slide and heat fixed. After heat fixing, you will add the carbol fission stain, which is usually pink in color. Once you add it, you will apply heat to the stain. So here application of heat, it acts as a mordant where the stain is penetrated deeper into the cell wall structure so that the particular bacteria gets easily stained. So after heating it for say 2 to 3 minutes, either you can use dry heat technique where you will directly heat the slide on a glass on a burner, or you can go for a steam where you will allow the water to boil and that steam is used as a mordant. After that, you will add the decolorizing agent, which may be your acid alcohol solution. Around 4 to 5 drops will be added so that complete removal of the stain, carboficient stain occurs. So here what will happen is when you'll be adding the acid alcohol solution, the unstained part or the non-acid fast bacteria who have retained that carbon stain will be removed by this decolorizing agent, whereas the acid-fast stain will retain this carbon stain. Then you will add the secondary stain, that is methylene blue, which is also called as counter stain. So while selecting the secondary stain, one has to take care that a contrasting color should be used, or a contrasting colored stain should be used, so that you can differentiate between acid fast or non-acid fast staining bacteria. So here, as you can see, we can either use methylene blue, which is in contrast with pink, or malachite green, which is again contrast with pink, so that you can easily differentiate between acid fast and non-acid fast bacteria. So when you will observe the slide under microscope at under oil immersion lens, the blue dots appearing are the non-acid fast bacteria, whereas the pink dots which appear, those are the acid fast bacteria. So in this way, by using this acid-fast staining technique or Zeil-Nielsen staining technique, you can easily differentiate between acid-fast and non-acid-fast bacteria. Thank you.